Hello everyone, I am Sir Remark and I am one of the teachers from the Biology, Earth and Life Science and Disaster Readiness and Risk Reduction cluster. In this video, I am going to discuss with you one of the most fascinating things the cells of our bodies can do. This mechanism is one of the most important processes that our cells do to sustain life. In this process, division actually means multiplication, wherein when something divides, it actually multiplies. And this process is called as the mitosis. This pie chart shows the portions of the cell cycle, and it is evident that the cycle is composed of two major portions or two major parts. The one on the orange or the one coded on orange and the one coded on blue. Let us first try to focus our attention on the orange portion. This portion is called as the interphase. The interphase is divided into three or more substages depending on the type of cell. These are the G1, S, G2, and in some cells, there is an addition of G0. These things are going to be discussed shortly. On the blue portion, we have it here. Okay, that's the cell division phase or the M phase, which stands for either mitosis or meiotic. I mean mitosis or mitotic, and then meiosis or meiotic phase, depending again on the type of cell that is dividing. Now, what this chart is trying to emphasize is the duration a cell spends in the portions of the cycle. Apparently, majority of the time of the cells are spent in the interphase. This is in the orange one. And interphase is also known as the preparatory phase. So majority of the time of the cells is spent in preparing for cell division. If they are going to divide. Now, this actually covers 95% of the cell cycle, this orange portion. Only a little portion is for the actual divisions of the cell. This means that cell division happens fast. Okay. Talking about cell division, as seen on the previous slide, this can be of two types. One is mitosis, wherein this division happens in somatic cells. Now, sir, what are somatic cells? When we say somatic cells, these are the cells that make up almost everything in our body, except the sex cells. So this means that they make up our skin, bones, blood, our different organs, and etc. as long as it is not a reproductive or a sex cell. Whereas the other type of division or cell division is called as the meiosis or meiosis, which happens in gamete-producing cells. When we say gametes, in common terms, these are the egg cells for females and the sperm cells for males. The sex cells are produced through a separate kind of division. Again, this is called as meiosis. But in this lecture, we are only going to talk about the type of cell division that happens in our body cells, which is mitosis. Now, again, in order for a cell to divide, it must first undergo interphase or the preparatory phase. Again, that covers 95% of the cycle. You can see it there on the screen now that the blue portion there is the interphase. So it starts from there. And then eventually, if the cell is ready to divide, it is now going to divide mitotically. So that is in the red portion. Now, as what I have been repeating in the previous slides, interphase is divided into either 3 or 4 stages depending on the type of cell. These are the following: G1 or the gap 1. Other references say growth 1, synthesis or the S stage, then growth 2 or gap 2 or G2 stage, and some cells also have this quiescent stage or G0. Now, let's proceed with G1. Again, these three, the gap 1, synthesis, and G2 happen in order. So let us first talk about G1. Okay, in G1, the cell is metabolically active and is starting to grow. When I say grow, it becomes bigger. So basically, it just increases in size. However, as it grows, there is no replication or when I say replication, there is no duplication or an increase in the DNA or the genetic materials of the cell. Basically, it just becomes bigger. Aside from that one, the cell's organelles are also duplicated to give the same number of organelles in the produced daughter cells. By the way, the produced cells after the division are called as the daughter cells. Okay? Now, after G1 comes next is the S phase or the synthesis phase. Now, if G1 does not replicate DNA, it is in this phase where DNA replication takes place or the doubling up of the DNA happens. This means that there is an increase in the amount of DNA of the cell because it doubles up. However, though there is an increase in the amount of DNA, the chromosome number does not change. No, sir. How does this happen? To understand why, let us first try to discuss about the anatomy of a eukaryotic cell. Aside from that one, we are also going to talk about the anatomy of a chromosome. Now, all eukaryotic cells' genetic materials are enclosed or encased in the nucleus. So the genetic material is found there. And this genetic material is the DNA. Now, this DNA is made up of two long strands that are attached together. This DNA molecule is tightly coiled in special proteins called as histones, in which later on you will see that one, to regulate gene expression, which make now tangled thread-like structures called as the chromatin. When these chromatins condense, they become more visible under the microscope, which are now called as the chromosomes. Now, most of you think or might think that the chromosome looks like this one. Yes, it is, but the chromosome may also look like this one. Now, that red one there is a chromosome with only one chromatid. And this red portion here is the chromatid. It is in the chromatid where we see the DNA. So that is the tightly packed DNA molecules. Then aside from that one, you are correct that the chromosome also looks like this one. This time, it has now two chromatids attached together at the central portion, which is called as the centromere. Now, these two chromatids are called as the sister chromatids. So, the composition of this chromatid is the same with the composition on the right. Okay. Once this chromatid replicates, or once the DNA on this chromatid replicates, it will now have a partner, which now they will which after that one they will be called as the sister chromatids. Okay. But again, this is one chromosome, and on the other hand, this is another one chromosome. Okay, aside from that one, and aside from the centromere region, the chromosomes also have this kinetochore, in which later on we will discuss this kinetochore as we proceed with the discussion on the cell division phase. Okay? Now going back into what happens in S phase, we have here an example. So let us say, for example, that the cell that is trying to divide is a somatic cell or your skin cell, for example. Now, every cell of the human body, with the exemption of sex cells, are made up of 46 chromosomes. So, if you are going to get a cell from your body and then um, dissect its nucleus, you will see there that there are 46 chromosomes. Now, again, that is sex cells are exempted for that one because sex cells only contain 23 chromosomes. Now, in S phase, there are 46 chromatids. If we are going to count these are all 46 chromatids. Therefore, we have 46 chromosomes. Now, after S phase, and again, S phase stands for synthesis. There is a synthesis of another strand of DNA molecule. Therefore, the amount of DNA or the amount of genetic materials increases. Then after that one, after the synthesis phase, we will now have 92 chromatids. So the chromatid now has a partner in which they are now called as sister chromatids. And in this case, we now have 92 chromatids. However, we only still have 46 chromosomes. Just a tip, count the number of chromosomes based on the number of centromeres. Once done in DNA replication or synthesis, comes next is G2. In this stage, proteins necessary for the actual divisions are produced, and that include now the enzymes. And aside from that one, the cell also continues to grow. So basically, those are the things that happen in G2. Now, for some cells, such as the nerve cells and liver cells, after G1, the cells enter G0 or the quiescent stage, wherein from its term, the cell becomes inactive for division. This means that the cell is living, so metabolism still happens. However, it does not prepare itself for division. Thus, it will not proceed or it will not enter S phase. Now, these cells will only enter S phase if there are cells damaged of the same type. For example, if there are damaged liver cells. So, liver cells will exit G0 to enter S phase until mitotic phase to replace now the lost or damaged cells. Okay? Now, the cell is ready to divide. It will now enter the mitotic phase, or it will now go to mitosis or to divide. Mitosis can also be called as the equational division, and this is called as equational division because the chromosomes are equally distributed to the daughter cells or to the produced cells. This means that the daughter cells produced after mitosis have the same amount of genetic material, and aside from that one, they also have the same number of chromosomes. And talking about the stages of mitosis, we will discuss that one shortly in the next video presentation.